The equivalence property basically tells you that if you build a function from one space to the other, this function is equivariant if it commutes with the group action of the input and output space. So for instance, if you have a segmentation algorithm, this segmentation algorithm would be equivariant if rotating the input image would also rotate the segmentation. That would be an example of equivariant function. And so basically, it's also pretty easy to see that if you have that for like two successive layers, I mean, it's a transitive property. So like if you have a first map that goes from 1 to 2 and a second that goes from 2 to 3, and both are equivariant, then the map that goes from 1 to 3 by composing these two functions is also going to be equivariant. And this is very useful in neural networks because you want this equivalence property to stay true throughout the whole network.
Welcome back, everyone. Today we have another guest, Vincent Mallet, who is a close collaborator and friend, but has some interesting work that we are excited to discuss. Thank you for joining us, Vincent.
Hey, Carlos. Thanks for inviting me.
Vincent, do you have like a quick intro of where you're at now? I know you're in Paris, but maybe you can give some more background on that.
Okay, yeah. So I'm a third-year PhD student in France, so in Paris. And I'm working at the Institut Pasteur, which is an institute mostly focused on biology. And I also have another supervision in Google and Lemin, which is more about the math. So yeah, and I'm working with like structural bioinformatics and trying to encode some priors about geometry and groups onto models that are designed for structural bioinformatics.
So if you're following the French PhD timeline, you should be done your PhD soon, right?
Yeah, in a year.
You'll do 4 years, or I guess you're starting your third year?
Yeah, it's probably going to be like 3 years and a few months.
Okay, I see.
Yeah, which
cool.
is the usual.
Wow, time flies. That's crazy.
Time flies.
Yeah, I mean, Vincent and I got to know each other when he was doing his master's in Montreal. So here we are now. All right.
Yeah.
So Vincent has a paper that got recently accepted to NeurIPS, which is very exciting. And I think we're going to focus on that. And whenever you're ready, Vincent, you can share your screen and show us your nice presentation. Also, okay. If you're okay, I think Perry and I usually will stop you if we have a question while as we're going, we can have a kind of open discussion. Great.
Yeah. Please
go for it.
Interrupt me whenever. Okay. So you see my screen and mouse properly?
Yep.
Yep. Okay. Okay, so today we're going to discuss this paper we got that's called Reverse Complement Equivalent Networks for DNA Sequences. And it's a paper I've written with my other supervisor at Lemin and Google, as I mentioned earlier, Jean-Philippe Vert. So as a little background on why we conduct this project is meant to deal with DNA data. So DNA is a molecule that most of you already know, and it's a polymer of nucleotides. So these nucleotides are basically little chemical groups, and they pair together. And there are only 4 types of nucleotides, and basically they encode in 4 bits the genetic information. And so this code is first translated into RNA, which I think is the focus of Carlos's work. And then this RNA is in turn translated into proteins. And RNA and proteins are basically molecules that explain most of the function of a cell. So they are really the main molecules, main focus of bioinformatics and biology, I would say. And so in terms of structures, this code is double-stranded, which makes it more stable. So A nucleotides always get paired with the T, and the G nucleotides always get paired with a C. And so this basically induces a redundancy, because if you're given a strand, so 1/2 of the ladder, you can deterministically know the other half of the ladder. And so that's coined as a reverse complement. And another thing that's interesting to know is that to access this genetic code, some methods, a set of methods were developed that are generally falling under the umbrella term sequencing. And that gives you access to reads. So basically, they are like substrings. So if you imagine the whole molecule of DNA, you have access to small reads. And there are always reads that basically are giving you information on one of the two strands. And the sequencing relies on biological phenomenon. And so it follows a biochemical direction called 5' to 3', and it's like basically a chemical direction, like it's based on the direction upon which the polymerization happened in DNA. And so if you were to sequence basically this like small DNA bulk that is here on the bottom left of the screen, you could get either this so ACA TTG result, or like the other way around, so CAT TGT. And so you can, as I mentioned earlier, deduce one of these sequencing results from the other based on the reverse complement symmetry or reverse complement action. And so for certain tasks, for certain biological prediction, you have a property that is bound to both strands. And I think a good example is transcription factor binding. So basically, transcription factor binding can be thought as if you have this DNA molecule that's floating around in cells, some other molecules might bind to both strands, to the DNA molecule as a whole, but actually it's going to bind selectively to certain regions of this code based on sequence complementarity. And so that's what I've depicted on the bottom as a teal line. So some regions have a higher affinity and some others have a lower affinity. And so, for instance, you might be interested in predicting these teal lines from the DNA molecule, and more specifically, because we don't have direct access to DNA molecule, you have to predict those from reads that I have here depicted as arrows. And because sequencing has like biochemical constraints, these reads, as I mentioned earlier, are like substrings, so they are short. They are randomly flipped because you can sequence both strands and you don't have a real control over that. And also they can overlap. So for instance, like these two arrows contain redundant information. And so,
yeah. Sorry, I don't think this teal line, I don't think I understand it. So this is like let's say the y-axis is the probability of binding or something?
Yeah.
And then the x-axis is the DNA, like along the DNA strand?
Exactly.
Okay, and then these sinusoidal waves on the bottom are what?
But that's sorry.
The sinusoidal waves on the bottom are just the DNA, I guess.
Yeah, that's it. Oh yeah, sorry. So like the DNA is this double helix. So like this sinusoidal represent the DNA. And
just a drawing.
The steel line would represent like the underlying profile of binding. So it's something that we don't really know, but there are some empirical measurements that basically let you know for a given sequence whether or not it's been bound to a transcription factor or molecule.
Right.
And so you get an indirect measurement of this teal line.
And the directionality of this blue and red arrow is just like the 5' and the 3' directions or something.
Exactly.
Okay, perfect. Thank you.
And when you receive the data from a sequencing output, you always get them in a specific order, right? Like from 5' to 3'.
That's it.
So the arrows are always kind of in the same direction, even though they were originally in a different direction in the DNA, I guess.
Well, I mean, no, because like the two strands of DNA are in reverse.
Okay, so sure,
I should have mentioned that.
Yeah.
So
Actually, so when
just the 5' to 3' is going on the left, and the red 5' to 3' is going on the right, and they are paired like that.
Yeah.
So
when I think we have a lag, sorry, but when the transcription factor, which is a protein, binds, I guess usually like the DNA first unfolds, no? Like the two strands separate. So does that mean like the same protein will have a way to bind?
No, no.
I mean, if I
So
remember
for
like my basic biology, you have these like they're called helicases, no? That like separate the two strands, and then proteins can come in.
Yeah, but this separation happens after the binding of transcription factors.
Oh.
So I mean, maybe some transcription factors also happen after these two strands get separated, but I mean, kind of the whole point of this paper is to address tasks where you have something that happens to both strands at the same time, and you don't know which strand was responsible for the binding, or which signal. I mean, and usually it's like a property of both strands at the same time. So like what happens actually, like biologically, to get like this data, like to kind of infer these teal lines from these arrows, is that you put transcription factor in the mix, basically, and these transcription factors are coupled with a part that is reckoned by antibodies, okay? And so first you mix the DNA with the transcription factor, so the transcription factor binds following the teal line profile, okay, in terms of distribution. And then you throw antibodies to the mix, or actually you coat a surface with antibodies, and you make the DNA bound to a transcription factor match these antibodies, and then you cut. So you get basically what you get is like a piece of glass, on it antibodies. Antibodies are bound to transcription factor, and transcription factors are bound to DNA. Okay, and then you cut like the DNA in small pieces and you wash. And so the only pieces of DNA that are left in your mix are the ones that were bound to transcription factors. So that's how the data like originates. I don't know if that was clear
Yeah,
at all.
and then you sequence the stuff that didn't get washed away, right? So you only sequence like the parts where there's a spike here.
Exactly. But when you do so, first of all, what happens is that, imagine here, I mean, maybe it's not the best positioning of arrows because it's actually where the affinity is supposedly low, but that's just a depiction. But here you would get, for instance, these arrows would probably be more in that region, right? Because that's probably where a transcription factor would bind. And so basically you would get arrows in this region of space. And then by sequencing them in a lot of replicates, you get kind of a Monte Carlo estimate of the underlying distribution of binding. Okay. And the thing is, like these arrows will overlap because the cutting is kind of random. So like you can have a few base pairs that differ, and also like the sequencing afterwards also is random. So you have like kind of redundant information. And so, yeah, that's basically what motivates the paper. Is that clear?
All good.
Okay.
I think it works.
Nice. And so basically now the underlying tasks for these kind of like applications is that you are given reads and you want to predict whether or not they will bind to a certain transcription factor. So to understand the relationship between like the sequence and the binding. And so to do so, there are like already some methods that try to include these kind of symmetries that your sequencing can be reversed. And so basically what they try to enforce is that if you feed as input to a neural network either a sequence or its reverse complement, you get the same like profile prediction or a binding prediction, because like it represents the same double strand molecule. Okay. But these methods were always kind of handcrafted, and so basically our paper tries to gather them into a common framework and investigate whether we have some kind of generality result on like, do we have any like all the possible layers that would satisfy this constraint that you want that your input and its reverse complement gives the same output. So to frame that into a maybe more formal way, we need quickly to introduce what groups are. So from a theoretical point of view, like the definition of a group is basically a set, and the elements of this set can be blended together by an operation. And this operation must satisfy a few rules that are basically that all elements can be blended one with another, and that when you blend two elements together, you can like reverse this blending and get either one of those. So concrete examples are, for instance, the addition of real numbers, for instance. So you can take two real numbers and add them, you get another real number. And if you start from a given number and add a certain number to it, you can subtract the same number and you get back the first one. So you have this invert operation for every element. And so you have the same for multiplication, but you need to remove the 0 because 0 times something doesn't have an invert. And you also have a very interesting group that is the invertible matrices, or subgroup of the invertible matrices, such as the rotation matrix or translation matrix. And so these groups basically can be thought as representation, because given an abstract group and a set of finite dimension, one says that a group acts on this set, on this space, by associating with each element of the group a bijection on the space, like from the space to itself. So a concrete example would be like, you can think of like rotations as an abstract group, or like you can combine rotations, like just we can discuss, oh, it's 90 degree and then 90 degree, but then you can make it act on a group of a given dimension. For instance, saying, okay, on a plane, planar rotation is going to rotate each vector following this precise bijection of the plane onto itself. And just as to give an example of how like this representation is not necessarily bound to a certain dimension, you could imagine also that rotations act on 3 dimensions by fixing a stable line, and then on the normal plane have the usual rotation act on the resulting plane. So basically you have group elements, and you can describe these objects with very theoretical properties. For instance, the modulo for real numbers would be the theta here, and you could describe everything that happens for rotations in terms of like the modulo group and the modulo operators. And so if you increase the angle and you go beyond the 360 degrees, you go back to 0. So that's like the structure of the modulo operator on on R, sorry. But then you can also think of how it acts on a given space, and that's more like the representation that we are used for groups, and it's deemed as representations, and it's like really what they are going to do to a given space. So, and then what? Yeah.
Sorry, I've never seen this notation, like this row goes from the group to a bijection. The way I'm used to thinking about this is that row, the representation, is a map from the group space to, say, some matrix space. So here, just so I understand correctly, this bijection here in this example of rotations is what? This map. It
would be one of those matrices, for instance.
So what's V?
Sorry.
So what's V?
So here, well, V would be a set. Here it's a linear space. Okay. And so for instance, here, let's imagine that V is R2, for instance.
Okay. So it's a biject. Okay. Yes.
Yeah, because you mentioned going from the group to the space of matrices, but actually it has to be invertible matrices for the second relation to stand, because you have to have an invert.
Yep.
And so actually the set of invertible matrices is exactly the set of linear bijections of V, but you can have more abstract.
I see. So your bijV is a set of invertible matrices. It's not.
Sorry.
Row doesn't bring. Row is not a map from the group to another map. Bij of V is your notation for group invertible matrices in 2D.
Yeah, that's it. I mean.
Okay. Sorry. Then I understand.
Bij of V is the spaces of bijections of V.
Okay, because when you say, yeah, that's fine. I think it's
as a concrete example here, yeah, you could replace badge of V by the set of invertible matrices of dimension 2 if V was of dimension 2, and if
I think it's
the bijections that you want to consider are only the linear ones. But you
that's
can
fine.
choose what.
I understand completely. I think it's just a matter of vocabulary. I think we learned this in a different area. All good. Thank you.
So basically, for each group member, you associate a bijection from a space to itself. So for instance, for a given rotation of angle 3, for instance, you associate the rotation that is going to perform a rotation around the origin of degree 3, of theta equals 3. For instance, that's like one way to represent your matrices. But you can also choose like other bijections for other auditors. For instance, you can choose to always put the identity here. So for every rotation matrix, you always put the identity, and it's a representation that's known as a trivial one, because I mean, it's really stupid. But then you still have that basically for each member of the theoretical group, you have associated an action on the space, and the action would be do nothing. And yeah, that's it. Okay, and so finally, the equivalence property is basically telling you that if you have a succession or a set of spaces, and each of those spaces are structured by a group action. So for instance, you have rotation acting on the input and rotation acting on the output. The equivalence property basically tells you that if you build a function from one space to the other, this function is equivariant if it commutes with the group action of the input and output space. So for instance, if you have a segmentation algorithm, this segmentation algorithm would be equivariant if rotating the input image would also rotate the segmentation. That would be an example of equivariant function. And so basically, it's also pretty easy to see that if you have that for like 2 successive layers, I mean, it's a transitive property. So like if you have a first map that go from 1 to 2 and a second that goes from 2 to 3, and both are equivariant, then the map that goes from 1 to 3 by composing these 2 functions is also going to be equivariant. And this is very useful in neural networks, because you want this equivalence property to stay true throughout the whole network. And yeah, that's basically the equivalence property, and that's kind of what we want to enforce. And a subclass of equivariance would be invariance, and it's a specific case where basically the output action here is a trivial one that I mentioned earlier, basically saying that you want the perturbation of your group to not affect the output space.
So in the case of segmentation, you wouldn't want that, right?
Yeah, you wouldn't want that.
Okay.
In the case of segmentation, usually you want the same group action on the input and the output, because I mean, like usually it's in the same domain. Like you have an image, and then you have a kind of an image also, but like with labels on it. And you can imagine other like fancier or weirder stuff. But more interestingly, like that's just based on like the input and output space. But in a neural network, basically, you're going to have the input space group action is going to be well defined. And then in the middle of your space, basically, you're very free to choose any group action and like follow the equivalence constraint. And then at the very end, you need to map back from a weirdly structured space with a group action that's kind of arbitrary to your output space. And so you have quite a lot of margin of action here, because in the intermediate layers, the group action is not constrained. Okay, so back to our initial problem. Now we can frame basically the symmetries that I mentioned earlier as group symmetries, and basically say that when you translate here, when you translate the arrow by randomly sequencing somewhere in the sequence, you are going to have some kind of overlap. And what you want is that if you overlap, like if you shift your input, so if you make a translation on the input, you want the predicting signal also to translate. So you have a translation symmetry. which is the usual one when you have convolutions, for instance. And there's an extra one that's the RC symmetry. And that is basically, it looks like the group Z2, which is kind of the smallest group, because given a sequence, you can get the other sequence by making some kind of like a flip. And then if you flip that flip, so if you take the reverse complement of a reverse complement, you get the input sequence. So basically, it's homomorphic to the group Z2. And so the resulting group action on the input can be decomposed into what's called a semi-direct product group. And basically, it means that our group elements are going to be composition of shifts and flipping, and that any element of the group can be decomposed as first choosing whether or not we flip, and then choosing or not we to shift. And it's a super intuitive result. Basically, like if you were to try and generate all possible perturbation of your input, you could just like either shift it or flip it. So it's that's what a semi-direct product intuitively is. A product group, sorry, is intuitively is. And then like it enables you to get and to leverage interesting like math results, such as using the induced representation. And basically what this means is that you can basically use a representation of the smaller group, so Z2, and then port this representation of the smaller group onto the group product, the semi-direct product group by using the induced representation, and you get a generative result that tells you basically that it's the most general way to extend your representation of the smaller group onto your representation of the whole group. Okay. And so this result of induced representation, what it means concretely is that when using those representation, the only thing that you have to choose is choosing a representation for Z2, which is a small group, and then you automatically have a way to get a representation for the whole group element. So basically, if you choose a way to represent the shifting action, then you directly and automatically have a way to find the group action for both shifting and flipping.
Sorry, Vincent,
Okay.
just to go back, I guess the operations that I had in mind were like flipping the bits and reversing the order, right? But now you're talking about like sort of a translation as well, no? Are those like three different things, or am I sort of confusing them?
So what do you mean by flipping the bits? Is like turning ones into zeros?
Yeah, to get the complement, right? That's what you mean by flipping, I guess. And then the reverse part is changing the order of the sequence, right?
Okay, so I mean there are two things here that I might have a bit. I think I was a bit fast. So like a first thing that can happen to a given strand is that it gets like you want to. It's possible that the representation that we have of a given double-strand DNA molecule, a bulk of DNA molecule, it can be either one strand or the other one. Okay, so that's the RC complement, and that's like a first thing that can happen. And at the same time, it's going to both flip the direction and also flip each nucleotide. Okay, in the input space. But another action that's distinct is that you could also imagine that you have sequenced two things that overlap, and here another perturbation that can happen to your input is that it gets shifted, and you want that when you shift your input, basically the output is also kind of shifted. So if you have like this profile annotation around this trend, if you were to first predict this one and then from the second red arrow predict this one, you want that in this region the two predictions coincide. Yeah. So these are the two subgroups that I mentioned, and then basically to generate all possible perturbation that can happen through your input, that's when you use the semi-direct product, where basically you say, okay, now like from a given sequence, if I were to, I don't know, like try and make perturbations over it, I could either like shift it and flip it, or flip it.
Yeah.
Okay, and so that's like a decomposition of what can happen to your input sequence. Okay, and basically what's going to happen on the input is more concretely that your matrices, basically, if you shift it, you're going to get zeros at the beginning and like lose information at the end, kind of. I mean, if you usually it's encoded as like an infinite signal, so like it would just like shift basically your signal on the line by saying, okay, I have shifted my input, so now codon position number 35 is now position number 23, for instance. So it has shifted. That's like the translation effect. And then the second effect is depicted here. And it's basically that if you have a given strand and the reverse complement happens, the order gets reversed and the bits are flipped. So these two things happen at the same time. And basically, then what I've mentioned about the induced representation is that you can actually represent this like double action of like shifting and flipping by decomposing it into something that happens at each position individually. Okay, so like here it was just like the bit flipping, and then something that's going to happen more at like the sequence level, where the order might get reversed and the sequence might get shifted. Okay.
Yeah, that makes sense.
So it's like, yeah, I agree, it's kind of weird because it's two decompositions that don't really like they closely follow each other, but they're not exactly isomorphic. I mean, they're not exactly the same ones, but like this, I don't know, like this shift is just arises from the induced representation and the fact that basically in the group action there's something that like the fact that you reverse the sequence is really something that affects the whole space, but the fact that you would flip the bits is kind of independent, so you can decompose your group action into like these two components. And so in the input, basically what happens is just like the intuitive stuff, where basically you flip your sequence, you optionally shift it and you switch the bits. But then what happens is that in the feature maps, as I mentioned, there you have no canonical way to define the group action, because basically it can be a bit arbitrary, right? And so
so in the feature maps here are like the intermediate layers of the neural net, right?
Yeah.
Like now, well, now you've mapped it to, okay, makes sense.
Yeah. So from the input space that behaves in the way I've just described under the group action, where basically the bits get flipped and the order can get reversed. From that like group action on the input space, you are going to map using neural network layers to map this input space onto other spaces. And there, to keep enforcing the equivariance, you need to define a group action also on these intermediate layers. Okay. And basically here, what I was mentioning about the induced representation is that then you can choose what happens to each bits. And so like you could replace flipping the bits, for instance, and permutating them, because like flipping is a bit weird way to see it. Actually, what happens is like you apply a permutation. So on the input, basically the A's become T's. So basically the first row and the last ones are permutated, and the second and third are permutated. So it's something that happens basically on each column. There is perturbation that happen, bijection that is applied on each column independently. And so this perturbation for the input space is just a permutation of like first and fourth and second and third. But then in the intermediate space, and the induced representation tells us also that it's actually the most general, you can choose other perturbation that don't really necessarily have to be anything special or like a permutation. And actually the most general thing that can happen and be a representation of Z2 is this formula. So basically what you're gonna apply on each column has to be a matrix or an operator of the following form. So basically you have an invertible matrix. Then on the diagonal, you have a part that's the identity, and then a part that's like minus the identity, and then the invert of the first invertible matrix. So what it means is that in those spaces, the group action is necessarily going to be in a given basis. Some axes are unaffected by the group action, and some axes are flipped. Okay, that's another way to see it, like more geometrical. And the permutation effect is for the regular representation is just when basically you have one sign that flip and the other that stays constant, and P is block diagonal. Then you get like that flipping the axis is going to turn this vector onto this one and this vector onto that one. I don't know if you see my fingers. And so it's just a way to tell you that like this action that we had of permuting channels one with another decomposes in a given basis into like leaving some dimensions unaffected by the group action and some others that get flipped. And that's a very general result. That's actually true for all representation of the group Z2. And so basically in the intermediate layer, yeah.
Sorry, here S IB is just the a matrix that's 1 for the identity and -1 for the other element of the group.
That's it. So like basically S is the group, the group element. So like because in the group, like formally you have the neutral element that basically does nothing. So if you don't like make a shift in your input, nothing happens. Never, because S is going to be 1. So this here becomes identity, and so the total stuff becomes identity. But when you have a flip in the input, or like when the group action is the one associated with the flip, then some more interesting stuff happens.
So basically, the simplest example, or a very simple example, would be P is identity.
Yeah.
IA is 1D representation. So it's a 1D matrix 1, and then SIB is 1 or -1. So you have those two group elements that are the identity in 2D, and 1 -1.
Yeah, exactly.
For the other element.
Exactly.
Right. Okay.
And so here, for instance, a vector in 2 dimensions that would follow the representation that you just mentioned would be like that, for instance. And when you apply like the negative element, the like first dimension would stay unchanged and the second would flip. Okay. Yeah,
so it represents a reflection. It represents a reflection around the y-axis,
exactly.
And fix the x-axis.
Exactly.
So if you reflect twice along y, you go back to the identity.
Exactly.
Which is exactly what you want.
Perfect. Exactly. And that would be like in the case where basically you have your basis vector are like that, so orthonormal, and you flip again around one of the two vectors, right? But now what I mentioned just earlier is that now if you choose to represent it into a second, like the same group action, but represented in another basis, so basically you change your basis vector to be those ones, and you do the same representation of shifting, going back up and down, then what you have is a permutation, because basically the first vector becomes the second one upon group action. So that's basically what I mentioned here, is that the regular representation, that is a permutation, is just one example where when you have A equals to B, so that's what we had before, because one dimension was stable and the other one changed. And P is block diagonal because, well, I mean, it's block diagonal because basically you choose the dimensions that you pair and you make the correct rotation so that your vectors fall in between the dimensions that doesn't change and the dimensions that changes. So that's just, I mean, it's not really to the paper, but it's just like to get a more intuitive way of understanding what can happen when you want to associate bijections to just the Z2 group, and you're pretty limited in like what kind of action you can have, and they all can be written like that. And so that's just like what can happen for a given column, and then using the induced representation, you can deduce the group action on the whole matrix based on what happens for each column independently. And so now what you have when you have all of that is that basically you have boiled down the whole possible group actions of this group that we have onto the intermediate feature space to choosing A, B, and P. So you just have two integers to choose and one invertible matrix, to choose for each layer at each feature map.
So Vincent, just to get like a really high level idea here, is the point of this to like describe what possible effects having these perturbations in the input will have on the feature maps? Like you're not actually constructing something right now that's going to guarantee some properties, right? It's just describing, it's descriptive, I guess, right? Okay.
It's kind of descriptive, but actually like this is a result, like a theoretical result, where basically you have all representations that decompose like that. So it's not just, okay, let's use this formulation and choose that. You have like an actual property that tells you the most general way Z2 can act on an intermediate feature space under this formalism is that one. And when you don't constrain A, B, and P, you have like basically all of the possible group actions on the intermediary feature space. So it's a bit more general as a result, but you are right that it is exactly about just picking what is going to be the group action effect on each feature map.
Sounds good.
Yeah. Okay, so now we have like kind of the mathematical framing of like what happens to our input and what can happen to like the successive feature map. And then basically we mostly rely on, I mean, all of that was kind of made to make us fall onto a framework that's actually a general theoretical framework. And that basically tells you, okay, if you have such a framing, you can always write mapping from one intermediate feature space to another intermediate feature space acted upon by such groups. And it all boils down to making a convolution with a constraint kernel. And basically the constraint of the kernel can be written like that. So the kernel with a group acting on the output times, I mean, like basically it's a formula. So basically you choose the group action row 2 and the group action row 1. So the input and output group actions, and then you have a constraint on your kernel. Okay.
By the way, I want to acknowledge the coolest first name I've ever seen in my life. The first author of this paper, Mr. Taco. I really have to hit back to group theory.
And basically, we re-derived also like this constraint in this specific example in the paper, but you can by setting a few stuff and framing it properly, kind of almost directly leverage this result that your function can be written as a convolution with a kernel that satisfies basically this equation. And then this equation can be also analytically solved for this group, the group that we have at hand, because it's a small group. And so you can like really find all the possible mapping from the feature spaces that we have discussed before. And basically what it tells you is that this kernel can be decomposed by block onto even and odd functions in the spatial axis. So yeah, so that's the result is also in the paper. And so, and it's also a layer that tells you like that all possible linear layers going from such structural space are can be framed like that. So basically, this is it tells you like what all linear layers look like. And then we also add another result about like the nonlinear layers. And basically, we derive the form of the most general pointwise commutative nonlinear layers. So pointwise commutative nonlinear are basically, I mean, pointwise nonlinear are the usual ones. So for instance, ReLUs, sigmoids, tan H, you always apply them pointwise. And what we mean by commutative is that given group action on the intermediate feature space, what we describe are when these pointwise nonlinear activations commute with this group action. And a side result of this theorem is that it shows that the regular layers are the most flexible. So the ones that were previously implemented in like kind of accidental like particular cases of this theory. I mentioned earlier that other kind of equivalent networks were handcrafted, and in this framework they basically relied on regular layers, and they are the ones that blend the most easily with non-linearities. But if you also use some fancier or different intermediate layer representations, you need to introduce specific functions, such as leaky ReLUs to be used with A kind or B kind layer types. And so in all cases, I mean, it's case disjunctions here, but well, long story short, given group action on an intermediary feature space, you can deduce the allowable class of functions that will ensure the equivariance gets propagated through the network. So yeah, long story short, basically what we've done is coin the problem of RC symmetry into the equivalence framework, because basically we want our results to be stable when we do a prediction over double-stranded DNA. And once we coin this into the equivalence framework, we were able to get all the linear equivalent layers that map from one layer to the other, to the next one in a neural net, and also all of the pointwise nonlinear commuting layers. And we've also implemented those layers in PyTorch and Keras, so you can build equivalent networks almost for free. And now the second question is it worth it? And we got state-of-the-art result using this framework, because I mean, the way we can think of it is that we have enriched the class, the family of function that was the equivariant family of functions that was available, because there used to be some equivariant functions that had been developed, but we have kind of unraveled the most, like the whole space actually. And so now we can tweak and try different members of this family. And we get, doing so, we have more space to tinker, which is usually very useful in deep learning. And so we see that basically when you look overall, so this is the performance on binding profile prediction. So the task that we mentioned early in the talk. And basically like the gap that we got compared to the former best equivalent method is approximately the same as the one that they had with using equivariance. So basically this is non-equivalent networks. This is one equivalent networks, and this is the best equivalent network in the family. And now a final word is that it's not actually a free boost. So sorry, this figure is a bit intense, but basically we unravel a whole family. And so if you are able to do some kind of grid search and explore the whole family of functions, and you have the time to do that, then you can always like kind of pick the best on the validation set, use it on the test set, and that's how we get like our best results. But the thing is, like, it doesn't seem that there is like one specific member of this functional family that always behave better than the others. And so it means that if you're budget constrained and you don't really have time to tinker a lot on a validation set, then maybe like by just choosing one, you will get suboptimal results. So that's basically what you have like here on the full data set and here on the reduced data set. For instance, the previous equivalent network, so what was here, the blue one, I mean, it's still the blue one. So we still have like the deep blue and light blue on this picture, and we still have that the equivariant is better than the standard. But then sometimes other equivalent members perform worse than what existed before, and sometimes they perform better. So like if you pick specific functions in this space, sometimes it behaves better than what existed, sometimes it behaves worse. And if you don't have time to kind of test it all on the validation set, sometimes we'll have bad surprises. So overall, I would say it's still a good, a very useful tool to use because it enables boosting the results, but it also means a bit more work because you have to tinker a bit and try stuff to get the best performance.
And
so
sorry,
the conclusion,
Vincent, back to the thing that when you write standard like as your first baseline, if I remember from the paper, that's not like completely ignoring the reverse complement phenomenon, right? Don't they like also sort of do a pre-processing step where they map the strands to their reverse complement and then feed it to the network or something like that?
Yeah, so basically the data augmentation. Yeah, so they train the network with like without constraining it to be equivariant, but like they use data augmentation to kind of try and enforce it, like make the network learn about this equivariance.
And is there any intuition as to like why that doesn't work? I guess you need more parameters or something. I don't know.
What's for sure is that, I mean, I don't think I have the figure, it's in the paper. Like if you don't use this data augmentation, it's like, for instance, here, like the performance I think would be around there, you know, or around here. Like you really have a huge boost by doing data augmentation. So it kind of works and it kind of learns to detect the motifs. But yeah, it's less efficient because I mean, I think the most straightforward answer is that when you optimize, it's probably easier to optimize in a constrained space. But yeah.
Yeah, makes sense.
Well,
yeah,
the question I have, sorry, I don't know if that answered Carlos's question. Yes. So, so this Spearman correlation, say I have no idea what this is. Sorry, but is this a significant boost? Let's say that the rightmost figure overall. Spearman correlation of 0.29 compared to 0.27. Can you frame that for me, just so I understand how much better you're doing?
So Spearman correlation is just I mean, you can abstractly think of it as like you have like very long vectors, and it gives you 1 if these vectors are the same, and 0 if they are completely intercorrelated.
Okay.
And so it's just a correlation measurement.
So this is like so it's 2% better basically. You get 2 if you had 100 entries in this vector, you get 2 more that are roughly the same.
I mean, there are some normalizations, so I wouldn't. I'm not sure. Sorry about like the computation. I must admit it's like a common metric, and I can't remember exactly how it's computed. So I wouldn't be able to actually tell you. But okay. I think like one kind of relevant comparison is more like you can compare like one method to another and look at the performance boost, you know. So for instance, like the best predictive model used to be like 0.25, and now it would be like 0.29. So it's like 4 additional %, but over 0.25. So it's like 16% better, I'd say.
Yeah, I guess I'm just trying to understand if it's like I said, is it 4 other things out of 100 total, or you know, is there also a normalization like you said that makes it so that it's is it a linear increase basically is my question?
I think not, but I'm sorry, I'm not sure.
That's fine.
But still, I mean, what you still have as a result is that basically this blue paper got published, you know, by beating the deep blue. So that's another way of saying it, you know. Like I think like for the community, like these are like kind of significant boosts to like be able to go from there to there was already something that kind of would help downstream tasks. So even though we are, I think, yeah, far from one and far from being perfect in terms of correlation, I think like improving the previous state of the art by 16% like of the previous performance is already kind of good. But yeah, I wouldn't know exactly what it means in terms of like how the vectors correlate because of the normalization.
That's fine. Yeah, there's no question. I mean, obviously you're doing better, but yeah, I just wanted to get a feel of like, are we? It's I think we understand each other. It's all good.
Yeah, and like here it's kind of the same. Like on these tasks, I mean, basically like these papers that take away are more like theoretical about like, okay, can it be worth it? but I think we have not put so much emphasis into the application and like what's actually going to be possible when using those networks compared to before, and is like the performance boost going to yield some kind of actual difference in biologists' lives. And I think we might actually do that. So I think in the discussion I talk about that here, for instance, like you see that on some transcription factors, like the values are like crazily high, like it's 0.993 and then it was 0.9925. I mean, like I think here like the relative performance boost is really tiny, so it's still significant if you we have basically conducted several independent training and compare the performance. So the error bars here are over replicates with different seeds, and so you see it's making a statistically significant difference to use these equivalent networks. But like the absolute performance boost, I think might not even be so relevant on that task. And yeah, that's one of the directions that I think we are going to pursue now, is to try and apply it to actual metagenomics problem and see if it actually can be useful for like tasks that are more maybe relevant. I mean, this I think is a kind of relevant task, but we have not conducted a so basically we have just used a subset of all possible transcription factors, but we have not tried training like yeah, on I think there are like a few hundreds of transcription factors, and we have not tried to investigate the performance overall for now. So you found my my third point, that was explore more applications. And as a quick conclusion, so yeah, we characterized basically the linear and pointwise RC equivalent layers and show that they improve performance. We mentioned already that some applications might be interesting to investigate a bit in a bit more depth. And as another open question that you have not picked is that in the nonlinear equivalent layers, I mean, I mentioned that we have kind of all possible networks, but actually that's not 100% true, because the nonlinear layers can be very, I mean, it's a very, very big space. Functions that are not necessarily linear, it's basically all functions. I mean, you can imagine crazy stuff where basically the nonlinearity is taking one number and mapping it onto 3 dimensions, or is taking as input several coupled dimensions, such as like capsules or stuff like that. So, and this we have, I think maybe there are like still some things to be discovered, and actually some, for instance, some capsule level nonlinearities are already in use for rotation. So, for instance, they look at like they have subspace that are not all of dimension 1. Some of them are of dimension 3, and of course you can apply a pointwise linearity on each of the coordinates of these 3-dimensional vectors, but some nonlinearities, for instance, use the norm of the vector, so it couples the 3 coordinates. And we have not looked into that because we have looked at the pointwise nonlinearity. So that's, I think, another perspective.
So is there like a general framework then, if you have some specific data set, you know that there are some invariances that you care about? Is it generally does it generally proceed as you characterize these invariances in terms of, you know, group actions, and then once you've done that, like that step of actually building the model that follows those invariances, is that sort of given already by this general theory paper? Like you just plug it into that and you get the model architecture?
So I would say that the few so if I frame it another way, like what's in my paper could be directly translated into doing it for like rotations in 2D, rotation in 3D, and so on. And that's basically work that the team basically of Max Welling is working on, I think. And I think it's getting more and more. So for instance, this was first developed in the context of 2D and 3D rotations. And then they made like a more general theory. And in a way, we could say that my work is just an application of this general theory, and I agree. I mean, I think I would be satisfied with that. I think the limitations are that you get this constraint, but it's not always easy to solve it analytically. So that would be like a first limitation. Then a second limitation is about like the nonlinearities. There is no theory on nonlinearities and admissible nonlinearities. that I'm aware of. So maybe very recent papers look more into that, but usually it's a blind spot of the equivariance theory. And then you always have some kind of like implementation challenge, I think. So when you solve, in our case, it was kind of, I would say, quite easy, because basically you use like convolutions that are well established in 2 dimensions, and you just have to set some kind of easy constraint pattern on the kernels. But for instance, in 3D rotations, you have to compute spherical harmonics, and this is costly and memory intensive. So I would say that's another possible like theoretical, not theoretical, but limitation of like just the theory. And then finally, like if you were to want to write a paper on like I don't know, rotations in 6 dimensions, then I think like it also takes a lot of time to do an actual validation to make sure that what you're doing is useful. So I don't know if you remember, Carlos, but my project as a master's student was actually using these 3 rotations, and I started by using equivalent networks that were already developed at that time. It was really early, I think, but it was already there. And what I found was that I couldn't make these equivalent networks converge correctly. So for rotations in 3D, it wasn't so easy to use an equivalent network and have everything work better. And basically, what's fun is that actually this paper that is cited here, so Zhu and Al of this year, is basically what we have mostly based our work upon. And so they were using this specific architecture, and then they were showing that other methods of enforcing equivariance, so it could be during training time to do data augmentation, but also what they were doing is some kind of model averaging, model ensembling, where they would do an inference on one strand and then on the reverse complement strand, and kind of make a group pooling over the results. So basically they get 2 outputs and they average them. And actually it turned out to be like super efficient. So in the big plot here, it's the line called post hoc. And you see that it actually outperforms any single model equivalent models here on this task, because we were not able to use the validation on this task because just for computational, for ecological reason, we didn't want to burn too many GPUs, so we just picked a few. And in this task, where we chose like the best equivalent based on validation, using just one equivalent networks was better, like it was a bit better than post hocs, but post hocs were better than the previous equivariant. So long story short, my point is that sometimes leveraging the theory is not enough, and you have to be a bit smart in like what layers you use and what prior information you encode. And it's not like just take any equivalent networks and it's going to be better than a non-equivalent network, or other ways to enforce equivariance, such as group pooling or data augmentation.
Is there a risk of like over inductive biasing it? Like could it be that sometimes like this what you think is an invariance is actually doesn't always apply, and you might want to actually like map the reverse complement to a different label from whatever reason, and then you just like miss all those points, right?
Yeah. So I think for a reverse complement, I think it's pretty limited. But for instance, for image rotations, usually like when you do data augmentation, you don't completely flip the images, because like a car is always sitting on its wheels. And so, for instance, that would be an example where for image pipeline analysis pipelines, it's not necessarily good to be completely equivalent to rotation.
I wonder if there's like a soft equivariance you can work with, no? Like some weird in between hybrid thing.
But in the car example, there's not 2D rotations available to you right in that plane.
There's no what?
There's no 2D rotate, like you said, there's a symmetry that represents all cars, say, I don't know. That doesn't include flipping it with the road under, like the road on top and the car underneath or something.
Yeah,
So you
but I mean, you can still take a bit your phone, and it's still going to be recognized, right?
Yeah, but I think
So like
I guess what I'm saying is the road is always underneath the car. So
yeah, but
so you can't.
But at the same time, like, not necessarily with a constant angle. So you have some kind of small rotation, but not to the full group. And here I think like maybe equivariance would fail to do that, because since it leverages group theory, you'd need to find a group that's like small rotations. But that
doesn't exist.
I mean, no, it doesn't make sense. But maybe you can think of like other group actions. So for instance, like the group of rotations could be represented maybe using other bijections, like I don't know, nonlinear geometry or stuff like that, where basically if you I don't know, imagine like turning, I don't know, line into like an asymptote or something, you know, so that like the great angle reflections just are mapped to small angle reflections or something like that.
Sure. Yeah, that makes more sense.
Yeah. There might be some kind of reparameterization that lead to that, but maybe it's not necessarily leading to improved performance.
Yeah.
But for some other analysis pipelines, equivariant works. So for instance, medical imaging, like since you take images of cells and cells don't have an up and a down, like that works well.
Yeah, so for highly symmetric data sets, it should work. Yeah.
Well, yeah, it's really what Carlos was mentioning. If your prior is actually true, like really true, then I think it's kind of always a boost. But yeah, sometimes it's limited.
I mean, bio is always super messy, right? I wouldn't be surprised if something ridiculous happened. Also, like the data itself is probably noisy, so you know.
Yeah.
But messy sometimes is good, right? Because you have all these like translational symmetries and rotational symmetries, and like, right? Because everything looks the same anyway you look at it, so it's always symmetric somehow.
Noise is very symmetric.
Yeah, exactly.
Very nice. I like that. Well, I think we all have next things to get to, and that's probably a good place to end, unless Perry, you have any other questions, or Vincent, any other points?
No, I'm fine. Thanks for having me, guys.
No, thank you. That was extremely cool.
Yeah, thanks for the presentation. Great work. Looks very cool. Have fun at NeurIPS.
Good luck. I hope this was like a useful practice or something. At least. Was this like a good practice for your presentation?
Yeah, I mean, actually, I had a presentation from NeurIPS, so it's more the other way around. But
yeah. You practice for this with NeurIPS. Nice.
There you go, NeurIPS. Take that.
Exactly.
All right. Well, thanks so much, Vincent. Hopefully, you'll come by Switzerland soon, eh?
Yeah. See you in Bâle.
Bye, guys.
Okay. See you guys.